We have a session for both 15 minutes to engage in questions and answers as you begin to think about the important ramifications of the announcements that you've just heard. And in addition, there will be a full panel discussion Thursday morning as well. So there will be more time to think, to engage in discussion and vice versa, both about the clean standards, joint project and partnership, the Clean Standards Science Committee, and others. On my left is a colleague who I have enormous satisfaction in working with and upon whom I rely regularly and very heavily, and that's Dr. Eugene Cole, who is Professor of Health Science in the Department of Health Sciences at Brigham Young University in Provo, Utah. And he has a distinguished background in having done very significant cleaning-related research with the Research Triangle Institute and others. I will let Gene elaborate, but he will be bringing back one of the most popular and important presentations from last year at the University of Nevada, Las Vegas, updated on measurement of cleaning effectiveness. Again, join me in welcoming Gene.
Thank you very much, Steve. I'll just say a few words to preface this presentation, this updated presentation from last year, and that is I had the fortunate opportunity back in 1990 to begin working with Dr. Mike Berry to do indoor environmental cleaning related research. And during a period of 7 years, we developed the concept of cleaning for health. And I can remember we decided we had sufficient information from the studies we had conducted in various locations across the country to begin to disseminate information. We decided to hold an East Coast and a West Coast seminar, 2-day seminar on cleaning for health. And we held the first one in October of 1997 in Raleigh, North Carolina. We had approximately 75 individuals, the majority of whom were from public schools, teachers, administrators, facility directors. We felt it was worth it to continue, and the following year, in October of '98, we held a similar and expanded 3-day meeting now on cleaning for health in Seattle, Washington. We drew over 100 participants in a format similar to what we have here. And again, the majority, over 70, were from schools. The area I work in, the broad area I work in, is public health. So in public health, we deal with reducing risks of morbidity and mortality in regard to populations. We have a vulnerable population. We have 50 million children entering school buildings of one type or another each day. We have significant absences due to chronic and infectious diseases. In fact, the leading absence, as you're probably aware, each day from public schools is asthma, which has an environmental component relative to various environmental triggers, often related to dust, also can be triggered by respiratory viruses and so forth. So this is definitely an area of great concern, of public health interest. We'll talk more, as Steve mentioned, on Thursday, more specifics about the standard. In that regard, I was involved in a number of discussions during our lunch break, and it was mentioned, you know, what we really need eventually is a cleanometer, and that we can take this cleanometer and maybe take it into a classroom, close the door, plug it in, flip the switch, it runs for X minutes, and if it's clean, a green light comes on. If not, a red light. I'm not sure we'll quite get there, but we've started the process, and so I make these initial remarks to let you know that we just didn't start thinking of this last year, or as ISSA has mentioned, even 2 or 3 years ago. But this goes back all the way to 1990. and a few studies even before that relative to the healthcare environment, which we've heard a lot about so far today. We have other related healthcare environments we haven't discussed yet. Daycare, which I have to admit, I myself have often referred to as seething pits of infectious disease. Nursing homes. We're still concerned with office buildings, with manufacturing facilities that require maintenance of ultra-clean rooms and manufacturing facilities. So what I'm going to address now, those of you that were here last year will recognize some of these. We have a limitation right now in terms of methodologies that we can use to give us information for an assessment potentially or a component of what we might determine as clean or a measure of cleaning effectiveness. That we've done something we call cleaning, physical removal, maybe there's another component, sanitization, disinfection, some type of treatment, that we visually say it is clean. What other measurements might we look at performing that can give us additional feedback and confirmation or validation or certification? So we're going to have to define our terms and use them carefully as the upcoming school standards research study continues. And we've already made great progress. We've identified a large school system. And again, I don't want to get ahead too much. We will talk about this more on Thursday morning, more of the specifics to bring you up to date. I would like to say, though, that before we start to look at some specific methodologies, the reason we need different types of measurement methods, be they quantitative, semi-quantitative, qualitative, is the fact that this refers back to the first presentation I gave here last year, and that is cleaning in special environments, that any indoor environment is an ecosystem. And there are microenvironments or compartments or subcompartments of that ecosystem. And in fact, when we look at what will be a final measurement or measurements of cleaning effectiveness in schools, well, we're going to have to look at that ecosystem. And we may have different sets of measurements to confirm or validate cleanliness. Why? Because we have different things going on in different compartments of that ecosystem. We don't anticipate we're going to find the same levels of dust, numbers and types of microorganisms, and other contaminants that we might monitor for in the classroom versus the cafeteria versus the restrooms versus the locker rooms, and so forth. And as we address different surfaces and materials, be they hard surface with settled dust, be they hard floor or carpeted surfaces. So it starts to become complex, but as we design research, then we take into consideration all of these factors and variables. I've been involved in studies where it has taken longer to design the study than it has to actually carry it out in the field. And it's critical that that be done. Why waste time? Why waste money? So what we're doing as a cleaning science standards committee, and we have for almost 2 years now, been collecting information, some of which you've seen today. Richard Shaughnessy and myself have done things going back, well, to the 1990s, and have been sharing information, collaborating now and then. And so we've been collecting information from various corners of the globe, putting it together. So we do have a base from which to start. And in terms of measurement of cleaning effectiveness, there are a couple of components here that are critical, and maybe levels or stages or phases. One, as was just voiced this afternoon, what can the individuals doing the cleaning, what can they do to measure how well they just cleaned, or in conjunction with the standards. So we're looking for some rapid methods that will at least give a very good qualitative or perhaps semi-quantitative assessment of, yes, they've achieved the goal in the classroom, in the hallway, in the cafeteria, in the locker room, in the restrooms. We have studies that have been published showing that some students, particularly in elementary school, junior high school, will not use the restrooms because they are filthy. There may not be toilet paper, soap, hand towels. They're often sites of violence, and we actually have statistics on children with bladder problems because they've held it all day long, refusing to use the restrooms in school buildings. So these are major public health issues. So it's the cleaning contractor or the in-house personnel who are doing the cleaning. What can we give them as a measurement tool? And then is there another, perhaps more sophisticated component that the contract manager or a QA specialist or someone who has ultimate responsibility for that building use as a measurement tool? So all of this to preface your introduction and thought processes various to a number of methods of measurement. And many of these you're familiar with. This is very general in the beginning. And I think this was mentioned today. There is no universal criterion, methodology, or technology for the measurement of clean. The goal of CIRI is let's define clean, and then let's work to develop methods to measure cleaning effectiveness based on whatever the practice is. So at some point, obviously, if we come up with a way to measure cleaning effectiveness, then that's going to be a tool that we can use to improve cleaning practices because we have a measurement device, a measurement tool. Why don't we have standard methodologies or technologies? Well, there's different types of clean. I just mentioned that due to differences in the ecosystem. This dictates quantitative measurement, such as dust collection, culturable viable sampling. All of these have been mentioned today. ATP, ergosterol, which is the main sterol that fungi produce, and has been looked at in terms of air sampling as possibly a measure of total fungal biomass in an indoor environment. It could be an entire building, it could be a classroom, and so forth. But it's a sophisticated methodology. It's not something the contractor can do. So, hospitals we've talked about quite a bit today, Acinetobacter, Clostridium. Typically, you've seen the data, it's collection of multiple wipe samples. They may be swabs, different types of wipe materials that can be used for isolation and identification of organisms. Cleaning effectiveness criterion may require a threshold of 0 from all samples. There's no way to ensure that all organisms are killed. Even with a disinfectant process where you have an efficacious product, it's being prepared and utilized in the proper way. And that's not our goal, and Dr. Gerber mentioned that. You know, we don't live in a sterile environment. We don't expect one. We want to lower the risk for inhalation, dermal exposures, and the like that might affect large populations. Sometimes we do want a perhaps 0 benchmark or threshold. For example, in working with IICRC and assisting restoration professionals over the years, I remember being asked to design a post-mold remediation protocol for a nursing home that had been flooded with sewage-contaminated waters. Patients had been evacuated, and of course these were the elderly who naturally are immunosuppressed. There were also cancer patients on chemotherapy, and there were AIDS patients and so forth, so very much at risk. They were removed to another facility. And then the question came up, you know, would you help us to design a post-remediation verification protocol? And so I said, well, it's a public health problem. and I'll work with you on that. And so depending upon the size of the facility, we came up with a design to adequately sample. There were several hundred samples throughout the facility at different stages of the remediation. Predominantly surface samples, minimally air samples. Why? Whatever's in the air came from a source, came from a surface material of some type. And so identified the numbers and types of samples, locations where they needed to be taken. And I put in one stipulation in terms of a criterion, wasn't the only one, but it was zero tolerance for Aspergillus fumigatus, which can be devastating to immune-compromised individuals, what we call thermotolerant. It can grow at body temperature. Every year it kills a certain percentage of individuals on cancer chemotherapy, those who've received transplants, and are immunosuppressed in regard to that and so forth. So out of all the hundreds of samples, no Aspergillus fumigatus, not even one colony, was allowed. If one was found, then that area or perhaps the entire facility would have to be recleaned. Whatever that meant, HEPA vacuuming, maybe use of a disinfectant or sanitizer, and then resampling again, and so forth. Didn't mean that there were no Aspergillus fumigatus spores there, but it's an indication that the risk of exposure has been reduced to where it hasn't been detectable in a random sampling design. So these are kinds of things that we need to consider as we do research aimed at developing criteria for measurement of cleaning effectiveness. We've talked a lot today about MRSA. Again, typically it's isolating the viable or culturable, as we preferably refer to it these days. And again, it might be depending upon, if it's an ICU, zero tolerance. If it's in the waiting room, okay, maybe we'll allow 1 or 2 or 3 CFU out of X numbers of samples. There are research tools, some of which we may use in the school standards study. Again, they're not methodologies that can be used by the cleaning contractor, the cleaning professional. One you see here, high volume small surface sampler. There's also a backpack version of this. It requires very precise attention to how it should be used. You can see square meters that are mapped off here in the other individual's hand. There's a stopwatch, and you have to move it so many seconds between the lines and so forth. But it gives a very precise quantitative measurement of dust per unit area of carpet. You can then analyze the dust for whatever you would like, viable fungi, bacteria, endotoxin, pesticide residues, and the like. Allergens, we've done that too. I've been fortunate to be involved in quite a few carpet-related research projects as we've looked at various antigens. So dust mite, mold, cat, dog. A big one these days in inner city environments is rodent proteins, rodent urine, mice, rat in particular, cockroach as well. So surface sampling, again, swabs, contact plates perhaps, as you see here, and then ATP, which we'll get a little more into in a moment. Surface sampling for residual fungal contamination. I mentioned mold remediation. Now, if I mention or you see a specific product with a brand name, we're not endorsing anything. It's just this is either representative of what's out there in the industry or it's the only one that's out there and therefore worthy of at least attention and consideration in terms of its capability. This is a Micrometer Test. I'll get more into that in a moment. It looks for a specific fungal enzyme as evidence of fungal mass contamination, whether it be a spore that's viable or not, whether it be hyphae, and actually a cleaning scale has been developed. This came out of the University of Copenhagen a number of years ago. The initial paper was presented at Healthy Buildings Conference in 2000. And it's interesting because representatives of the company, I talked with them at Healthy Buildings, and then they came over twice to the US and each time asked me my opinion of the test method and what about application to post-remediation, fungal contamination, that type of thing. And I said, you know, I've looked at all your materials and your data, and I'm more excited about its application for measurement of clean and cleaning effectiveness. And they said, well, you know, we've got to get a return on our investment here, and we've got to sell these things right now. And I said, well, I'm just not interested in that right now. And, you know, I was interacting with CIRI, and there were thoughts of a mold remediation standard, which we're just about ready to go to press with the second edition. But it still potentially can be used for that purpose, and I'll show you some information. Bacteria, Stephanie Dancer showed you some auger slides. There's the round contact plates, they work the same way. Petri film, you can take a swab, then emulsify it, suspend what's on the swab, and then inoculate your Petri film. And in 18 to 24 hours, you have selectively, if need be, a quantitative measurement of gram-negative bacteria, maybe specifically coliforms, staph. So very handy, very useful. Again, not something that perhaps the cleaning professional would use. Maybe the cleaning professional is going to use ATP in addition to visual assessment, but then maybe they'll take something like a contact auger plate or slide, and then maybe even something, a swab that's processed with a Petri film for additional confirmation 24 to 48 hours later. As we talk about, and I show you some ATP data, we are going to interpret those, and we're going to consider hotspots in environments. Again, let's think schools, since that's on our minds. There are going to be hotspots, okay? We would expect to find less bacteria, for example, vegetative bacteria, adhering to wall surfaces, okay, 2 feet above the baseboard than we would at the level of the students who are going down the hall and touching the walls and the handrails and that type of thing. Same thing, you know, 2-3 feet down from the ceiling. So we would expect a gradation, and that has been demonstrated. I'll show you some specific ATP data in that regard. We can get very sophisticated. We haven't really talked or discussed yet, even in our Clean Standards, Clean School Standards Committee yet, in regard to microscopic examination. Carpet professionals, okay, always have a little hand microscope that they can actually observe carpet fibers before and after carpet cleaning, look at wear, whether a carpet needs to be replaced. Is this some qualitative measure that we could also use for not only carpet and fibrous materials, but perhaps hard surfaces as well? So these methods can be summed up saying that there can be some great technology here, but very complex, requiring highly technical skills, labor-intensive or time-consuming, also not necessarily cost-effective, lacking an acceptable framework of standardization, which we hope to clear up, not suitable for rapid routine cleaning effectiveness use. Some more of those. Again, just to give you some background and have you understand that a lot of the data that have been generated out there from good research studies that have been published, presented at various ISIAC meetings and the like have involved methodologies that aren't user-friendly. So the HVS3 that I mentioned, great information, difficult to use. It's This happened to be a Royal vacuum retrofitted with a cyclone. The price at the time was $3,000. This isn't something that's going to be in every school to use. The BM dust detector, Richard Shaughnessy mentioned that. Don't try and go online and find out where you can buy one, because they're still manufactured, but it's still mostly a research tool. It involves clear gelatin foils that are put on a surface. This standardized size. Comes with a little rolling pin. You have to apply a certain amount of pressure, no more than 1 pound of pressure. But then you peel it off and you put it into a photosensitizing device that basically gives you a measure, a spectral measure of dust, which would be blocking the light waves. For research purposes, it's yielded important information. Ergosterol, as I have here, requires a gas chromatograph, or at the very least liquid chromatography. It's a research tool. Endotoxin has been mentioned today, and it's a component of the cell walls of gram-negative bacteria. So these are soil bacteria. These are coliform bacteria. In fact, we've had a lot of mention of these today. Acinetobacter is gram-negative. E. coli, Pseudomonas, we can go down the list. There is no rapid method. You can collect samples. You can turn around your samples in a day within a laboratory that's very efficient at doing this, but it's not cost effective, and it's hard to interpret. So, and I think it was Dr. Gerba that had varying measures of endotoxin units on airplanes. The carpet versus the seat versus another surface. We don't know what that means in terms of human exposure and potential resultant health effects. So we're down to methodologies that are somewhat sophisticated, requires use, experience, but can give us important information, maybe in combination with 2 or 3 plus visual assessment. So the bioluminescent testing, looking for ATP, which has already been defined. It's the energy driver for all living cells. So it's in our epithelial cells that we shed. It's a component of bacteria. Anything basically that came from a living entity and that's organic. Enzyme testing I mentioned. So let's talk a little bit about ATP. It is I still classify it as a semi-quantitative method. There is a scale, again, that we have to interpret based on how we're going to use it. Many of you use ATP in terms of the work that you do as a verification for your own information. When I talk about the micrometer, there are some mold remediation contractors that are using it for their own purposes to see how well their cleaning is doing before they decide the job is complete. This is the Firefly Interaction, luciferin, luciferase. So it's a photometric determination. Collect a swab, and there's several different brands on the market. You then squeeze the tube, you get the chemical reaction occurring, and in 30 minutes or less you get a reading of lumens in a luminometer, or we might call it something else. Background materials can vary, and I'll show you some data in a moment. When we initiated a study going back several years now, we found natural background levels, which you have to factor in. They're always there. For example, we looked at wallboard that was not painted and wasn't wallpapered, okay? It just had the paper facing on it. Well, that paper contains ATP. If you take absolutely clean wood and take your swab and do an ATP reading, you'll get a measure, usually, of some type. Why? Well, wood is organic. It still has ATP, whether it's bound or not. But if we're going to make a determination, have we improved cleaning. We improved surfaces and reduced contaminants on walls. There's wet mopping. Here's cleaning of light fixtures and so forth. Well, we need a baseline to interpret what those results are now than what they were before. And we need lots of baseline information from thousands of surface sites, perhaps in hundreds of different buildings. This is actually at the end of the slides that I was working with there in that section of your binder. Some work we did a number of years ago for Joe Arrigo, who's here with us today, cleaning professional, very much involved at that time, 1999 to maybe 2001, 2, in wanting to know how effective was ATP, how could it be applied to various aspects of cleaning, surfaces, materials, post-remediation, restoration, that type of thing. We started off with purchasing pure ATP and inoculating different materials, as you see here, wet and dry glass, wood, which has its own natural background level, and then wallboard. Main thing I want to point out here in terms of pure ATP, as you can see on the horizontal axis, in terms of picograms, we begin to pick things up at about 10 picograms. How does this relate to what you read on the meter? Depends on what meter you have. You know, they're not all standardized. So again, does it really matter? Probably not, as long as you can derive an interpretation with your ATP device that is equivalent to someone else's who would sample the same surface and maybe get a different reading in lumens and so forth. That's the key. How does your luminometer reading relate back to amount of ATP? Now, this is E. coli, so there's going to be ATP there. And the key point here, if you'll notice, we could not detect any ATP until we got to a concentration of 10 to the third, which is 1,000 cells in particular. We very specifically quantitated the inoculum. Is this a problem? Well, on surfaces and materials, probably not, unless it's in the school cafeteria or a food processing facility and that type of thing. But so we have 10 picograms of pure ATP, 10 to the third of bacteria that we have a concern with on different materials. And you can see we have linear curves, which is nice. Here we used a yeast, same thing. We still have wood can be all over the place just because it has can have considerable background ATP. I know that luminometers today can adjust for this. At the time, the particular device we were looking at showed a rollover, and that is if ATP is so high, it actually goes to the maximum on the scale and then rolls back over to zero and starts over again. So is that important? Well, if you're doing pre-cleaning, probably not, as long as you realize that if you have a zero reading or very low, it's probably been in a rollover phase. Natural background, ATP, well, here's just glass versus wallboard and wood. And this is just a spectrum of materials that we sampled all over the place. As you can see, wallboard and wood, the range is 0 to 100-something thousand lumens, which means there are varying degrees of ATP on surfaces. What we did was we went into 3 different environments. One was a school, one was a dental laboratory, and the other was an office building. And we took samples from the baseboard to the middle of the wall and then up near towards the ceiling. And you can see just with these few samples, this was preliminary, what we have. We had baseboard vinyl. We have a range, 2,000 to 695,000. Well, how are things being cleaned? And people are mopping floors, and so you can get a buildup of residue on baseboards if the baseboards aren't cleaned after the floor is cleaned. A painted wallboard from 0 to 55,000. And again, I can remember in the schools when the samples were collected, obviously we got higher values where the children would go down the hall, changing classrooms or going to the cafeteria and that type of thing. So there are gradations of ATP relative to hot spots, and we would hope to identify those as we do the school standards research and then make recommendations for post-cleaning effectiveness measurement based on identification of those hot spots. Classrooms, cafeteria, locker room, restrooms, that type of thing. Finished wood, painted metal. Again, if there's an organic component of some type, we're going to get an ATP reading. Or even if it's not organic, if people are touching it, okay? If that painted metal is a handrail, then yes, you can come up with levels of 49,000 on your luminometer. Really not made for fleecy or fabric materials, but hey, we went ahead and did it anyway. And again, spectrum of ranges, obviously. These materials collect dust. Dust contains a variety of proteins, allergens, pollen, bacteria, and the like. Let me switch back now to what I was talking about previously. The Micrometer Test, this is an assessment for a very specific fungal enzyme that you see there, N-acetyl beta-D hexosaminidase. It doesn't occur in bacteria. It's specific for fungi. So in that sense, it's more of a targeted, if you will, mechanism, not mechanism, but indicator, perhaps of contamination. And so this involves collecting a swab, using a template of so many square centimeters, and then exposing that to a solution, adding a reagent, and looking for a color change that's measured photometrically. There you see the template. That was an older form of the photometer that was used. The research that was done at the University of Copenhagen determined acceptable levels of cleanliness, and they categorized them as A, B, and C. So this sounds simple. Maybe we're onto something here that could easily be done by someone who's actually just completed their routine or maybe a specialized cleaning activity in a certain building, let's say a school. A would be background level, mold spores that have settled on a surface or somehow adhered, but they're not actively growing. There are no hyphae. We expect low levels of fungal enzyme. B, it's elevated due to settled dust. Remember Richard talking about settled dust, and that his research data has shown if you have hard surface floors, you will have more settled dust in school classrooms on the shelves and on top of file cabinets and so forth. Why? Because the floor dust is more easily resuspended. It has to go and settle out somewhere. So we're talking about settled dust and higher levels. Why? More spores have settled out there. C would be the highest level, and this time most likely indicative of active fungal growth. There's a moisture problem. So we begin to use our protocol when that's finalized in the Clean School Standard study. We're going to have to characterize those buildings. Do they have water intrusion, moisture, mold problems, and the like? We have to understand that ecosystem. But this is what folks at Micrometer and the university came up with. Category A, less than 25. This is based on their baseline data. They just went around to buildings all over Copenhagen and collected information, schools, office buildings, healthcare facilities, so forth. I kind of like this. Is this the end-all and be-all? Probably not. And again, I doubt that we're thinking of one or even two particular forms of assessment that we need to consider in terms of cleaning effectiveness. Certainly visual, but then two, maybe three methodologies. Is this one of them? Don't know. This is why we need to do the research. C would be over 450, and this is based on, again, the numerical readouts that you get with that particular type of photometer. Here's one that's being used widely in the food processing industry, rapid protein residue test. Okay, so maybe this is an additional component. So we'll seriously consider this as we design our study protocol for the schools. Total protein, yeah, will that work in conjunction with ATP and maybe the fungal enzyme and maybe some culturable bacteria and visual assessment? You know, is that what we're looking at? Again, we don't know. This involves a swab, again, a specific area, let's say 4, 8, 10 square centimeters, goes back into the tube, you squeeze the top, the reagent goes down to the tip of the swab, and in less than 10 minutes, you observe a color change. It will turn purple if you have protein, and the degree of color change equivocates to the concentration of protein, the amount that you have there. So is this an adjunct, perhaps test? And again, we also have to consider what are these going to cost? And we haven't really gotten to the economics yet of validating, verifying, certifying, cleaning effectiveness. It's going to add to the cost of these services. But we hope that as we begin to, or continue, I should say, disseminate information as a professional cleaning organization, that we will educate and thereby empower parents who are concerned about their school facilities to demand that the schools be clean. They know their children get sick. We know we have children out whose asthma has been exacerbated and so forth. So what do we need to do? A lot of comparative research to identify optimum cleaning effectiveness measurement methods for, where it gets a little complex, but we have to do it this way, specific target markers across different environments, so, or subcompartments of the building ecosystem, materials and surfaces, so painted wallboard, tile floors, metal handrails and the like, and applications, what's going on in those different environments. We know from today's presentations that we're doing things differently, we're measuring things differently in the ICU in the hospital than the outpatient clinic environment. So we're doing this, the second bullet, we're doing all of this in a cooperative mode of effort to define the many aspects of clean. There's not going to be a universal clean for every service in every environment, for every application. So we've got a long road ahead of us, but we're on that road and we're picking up speed. And as has been said by Jim Harris and Steve Spivak, this is a cooperative effort. We would like your input, feedback. If you know of other methodologies, if you've been using one for several years and it's worked for you and you know how to interpret it, and doesn't impact cost of what you do in providing cleaning services, let us know. Share your data, okay? We want to benefit the entire industry and then ultimately the health of the public. Thank you very much.